All right. Good morning, everyone. Uh, yeah, welcome to Biocham 315 class continuation. Today we are going to talk about amino acid degradation. So, just like we discussed the last class, um, we talked about uh, bed of amino acids, and after consuming or eating food that contain amino acids, several things happen, and finally lead to absorption of this amino acid into biological system. And this amino acid could be used basically for major processes which we mentioned, and one of them is that it is used for the synthesis of protein. If all the amino acids needed for the synthesis are enough, and you have excess of this amino acid, and the body may also need to do other synthesis, like the synthesis of nitrogen-containing compounds, which will be discussed in the synthesis part of this class. And another thing is the excess that you need, the body does not need, cannot be excreted. Rather, the body will convert it to another substances. Could be for energy production, or if the body is in a state where it doesn't need energy, then the body will convert it to either fatty acid or a glucose, which can basically be converted to a lipid or triacylglycerol for storage, as well as a glycogen for storage. So we have several of these amino acids. All the 20 amino acids can be converted to about 6 major compounds. These major compounds are either metabolites of citric acid cycle or uh, other compounds that can easily be used for the synthesis of glucose. For example, here, based on this diagram, we have 5 amino acids can be converted to acetyl CoA, and we have 4 amino acids can be converted to acetyl CoA directly, and we have 6 amino acids, 5 amino acids can be converted to pyruvate, 2 amino acids to oxaloacetate, 2 to fumarate, 4 to succinyl CoA, and we have a 5 amino acids also that can be converted to alpha ketoglutarate. So basically, these amino acids are classified into two. It can be ketogenic or glucogenic. They are called ketogenic if what they are producing will end up producing either a ketone body or a fatty acid, but they cannot be used for the synthesis of glucose. Whereas the other class, called glucogenic amino acids, can be used for the synthesis of glucose. So basically, the one in blue that can be used to produce acetyl-CoA and acetyl-acetyl-CoA. Acetyl-acetyl-CoA can be converted either to acetoacetate, which is a ketone body, or can be converted to acetyl-CoA. And once acetyl-CoA is formed, the acetyl-CoA can enter a citrate acid cycle when the energy is needed. But when what is needed is not the energy, then the acetyl-CoA can be used for the synthesis of fatty acid. Likewise, also, those that were converted to pyruvate can be taken into citric acid cycle. We know the reaction of pyruvate. It could be pyruvate dehydrogenase to acetyl-CoA when the aim is to produce the energy, and then the citric acid cycle continue. But if the aim is to produce glucose, then the pyruvate carboxylase will be converted to oxaloacetate. Then oxaloacetate we know can be converted to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase, and this reaction is one of the bypass of the irreversible reaction of glycolysis. So you can remember the point where energy is produced are mostly uh, the points where the reaction of a glycolysis is irreversible. So when you go down and produce phosphoenol pyruvate can go back down to the reaction of phosphocryptokinase 1, and there is also a bypass, and then finally the reaction of hexokinase. So you can have phenylalanine tyrosine converted to fumarate. You can have isoleucine methionine proline converted to valine. You can have arginine, glutamine, histidine, proline, and glutamate converted to alpha-ketoglutarate, which all can be converted to oxaloacetate. So oxaloacetate doesn't need to form acetate with acetyl CoA because the excess of these things, or the aim of the production of these things from the fatty acid, is to produce glucose. Therefore, oxaloacetate can be taken out and then produce phosphoenol pyruvate, which eventually lead to production of glucose, and the glucose can be used to produce glycogen. We'll see some of these reactions. Okay, so before we talked about, just like we discussed in the last class, we talked about some basic reactions in which we'll be discussing throughout our discussion in amino acid metabolism. And one of the reaction we discussed is transamination reaction. In transamination reaction, we will explain what transamination reaction is. The next one is so, as I was explaining, when you have